A lot of my work is focused on regulatory T cells. These are a small population of CD4 cells endowed with very potent suppressive capacity. They play a key role in preventing autoimmunity, inflammatory disease, so they play a key role in modulating and maintaining a healthy immune system. Any person that has a defect in regulatory T cells, such as the IPEX, will get a spontaneous autoimmune disease, so they're really important cells. But by the same token, patients that have IPEX that receive a bone marrow transplant completely restore their immune function. So they both have significant consequences if they're not present, but also great potential restorative capabilities if restored. Unfortunately, even though they play this key homeostatic role, they are upregulated or enhanced in a number of diseases, particularly in cancer, and therefore they represent a major barrier to effective anti-tumor immunity. That's been demonstrated in mouse models of human cancer, and a lot of clinical trial data showing correlates between increased regulatory T cells with either poor prognosis or poor response to some therapeutic treatments. There's obviously enormous interest and excitement about the efficacy that's been seen with checkpoint inhibition. Anti-CTLA-4 and anti-PD-1 are obviously in the clinic. And of course, there's inevitably a lot of attention to additional checkpoint inhibitors. LAG-3 was the next one in the clinic. Actually, that's something that we have particular interest because we've been working on LAG-3 for over 15 years, probably published more papers on LAG-3 than most other groups. So we're very interested in that. But clearly, a lot of people are thinking beyond checkpoint inhibition. Obviously, people are interested in trying to upregulate co-stimulation through agonist activation. But with regard to the focus of my talk and what we're discussing here, people are also trying to find ways to manipulate regulatory T cells as well as other inhibitory populations. And that's further behind with regard to clinical development, but there are 3 or 4 ways one could think about modulating them. Firstly, by modulating their capacity to migrate into the tumor. And indeed, that's probably the first thing that's in the clinic. A molecule called CCR4 is a chemokine receptor. It basically functions to guide a cell where it migrates. And a lot of tumors will make high amounts of the ligands for CCR4, therefore attracting regulatory T cells into the tumor microenvironment and thereby helping to protect them against the immune system. And so an antibody to CCR4, mogamulizumab, is in clinical trials at the moment. Too early to say how efficacious it'll be, but that'll be interesting to see if that is an effective modality. A lot of academic groups and companies are also developing therapies against either effector mechanisms or suppressive mechanisms that regulatory T cells use, or other pathways that might generally undermine regulatory T cell function in a selective way to obviously not impact the tumor-specific T cells. And of course, the other important thing about any therapy that targets regulatory T cells is it really has to be relatively restricted to the tumor microenvironment, because obviously a substantive effect on the general regulatory T cell pool may induce autoimmune or inflammatory disease. So it has to be quite surgical, and a lot of efforts are going in that direction. I think in the next 5 to 10 years, hopefully sooner, we'll get a sense of whether that's an effective strategy to combine with all of the new developments that are coming along the pipeline for cancer immunotherapy. There's almost a bewildering number of combinations that are being tried at the moment, and certainly people are trying to combine checkpoint inhibition with conventional chemotherapy or radiotherapy. I think the kind of general goal is, or sort of mindset, is to not just simply combine things that are similar, although that has shown to be efficacious. So PD-1 and CTLA-4 are both checkpoint inhibitors, but have shown efficacy by combining. But I think most people believe that if you can combine therapies that target things in quite different ways, that will be efficacious. So there's certainly been efficacy seen with checkpoint inhibition plus chemotherapy and also radiotherapy. And we shouldn't also leave out vaccination. DC vaccines have been in the clinic for a while, and those are being combined with checkpoint inhibition. And very recently, people have become excited about the potential of oncolytic viruses to not only target the tumor and modulate the tumor microenvironment, but an opportunity to deliver so-called payloads that could modulate the immune system further. I think it's suffice to say that regulatory T cells are an amazingly exciting population of immune cells endowed with very potent properties that could be either targeted for therapy in cancer, but of course we shouldn't ignore the other side of the equation, which is that they are obviously being studied extensively to see how their activity could be enhanced to treat autoimmune and inflammatory disease. So a lot of the research is being done to focus on understanding these pathways in cancer, particularly because regulatory T cells and inhibitory receptors are most upregulated in cancer, could have beneficial consequences far beyond cancer in terms of deriving therapies for many other diseases.